Sirugo from the University of Pennsylvania School of Medicine. The title of my presentation today is The Missing Diversity in Human Genetic Studies and Its Impact on Health. For the field of human genetics, the past 20 years have been characterized by fast discovery, impressive technological development, and also notable cost decrease. We went from mapping the human genome through a complex international collaboration, which took quite a bit of time to be concluded at a cost of billions of dollars, to the current ability which we have to sequence individual genomes for a tiny fraction of that original cost, just $1,000 per genome, in a short time. Nevertheless, in spite of all the progress made, some key challenges persist and some key questions remain unanswered in the field of human genetic research. How do ethnically diverse humans differ in regard to genomic and phenotypic variation? What are the evolutionary processes generating and maintaining that variation? How do gene-protein, and gene-environment interactions contribute to phenotypic variability in humans? How does all this impact disease risk? Now, patterns of human genetic variation are extremely complex. This complexity is the result of the long evolutionary history of modern Homo sapiens. To make the long story short, modern Homo sapiens originated in Africa. The earliest fossil evidence dates back to 300,000 years ago in North Africa. Africa is the continent which has witnessed for the longest period of time evolution of modern humans on the face of this planet. We also know that approximately 100,000 or 60,000 years ago, a group of humans left Africa in what is called the Out of Africa dispersal, a major demographic event. And they carried along with them, they carried their gene variants. And since what left Africa at the time was just a subset of all possible variation existing there, that explains well why the highest level of diversity is found in African populations and not elsewhere. In addition to the large amount of genomic diversity in human populations, which I mentioned before, we also know there is a lot of phenotypic diversity. The incidence and prevalence of human disease change across ethnic groups. Here we have the case of high blood pressure, highly prevalent among African Americans with respect to Mexican Americans and European Americans. The prevalence of diagnosed diabetes also changes by ethnicity, highly prevalent among Native Americans, as well as African Americans with respect to European Americans or people of Asian ancestry. And here we have a very interesting correlation between the percent of ancestry and risk to have a common disease. This is percent of African ancestry correlating with risk of having type 2 diabetes. And a very interesting conversation to have is, if there are gene variants involved in determining this correlation, are these variants only directly involved in biological processes determining diabetes, linked to pathogenic mechanisms determining diabetes, or is there something else there which is still genetic but not directly involved in biologically involved in causing disease and yet linked to ancestry? For example, genes determining skin color, this is a very important conversation that we should have. And then finally, analyzing comorbidities by ethnicity can be particularly complicated. We see here the combination hypertension, hypercholesterolemia. We do not see much difference between African Americans and Americans of European ancestry. But if we look at the comorbidity hypertension, diabetes, differences are striking. And there are many examples like that. The analysis of comorbidities by ethnicity is complex, and there's still a lot of work that needs to be done in that area. Cancer is another area where we observe strong disparities. Lung cancer is a leading cause of cancer mortality in the United States. It has the highest incidence among African American men compared with other ethnic groups. Even considering similar exposure to the carcinogen type, the rate of lung cancer differs by a factor 2 to 3, according to self-reported ethnicity. Of course, when we run this type of study, we have to consider many factors, environmental factors, behavioral factors. That could be, for example, the type of cigarettes smoked, or the so-called smoking topography, path volume, smoking duration. Or it could also be low uptake of pharmacological cessation aids among subjects belonging to a certain ethnic group, and so on. It is possible that all these factors also combine with some genetic or epigenetic factors contributing to the disparity. We really do not know much about this, and a lot of work still needs to be done in this area. Now, for the past 15 years, since 2005, when the first GWAS was run, a standard way to investigate the genetic basis of common diseases has been by genome-wide association studies. In these studies, millions of variants are typed at the same time in thousands of individuals to see if a variant or a group of variants associate with a trait or disease of interest. In the recent past, Alice Pope-Joy, Alicia Martin, and others, along with Sarah Tishkoff, Scott Williams, and myself, have highlighted a problem, which is that the great majority of these studies is run in European populations. There is a very strong bias for this type of genetic studies, and it's very, very hard to transfer genetic findings from European population to non-European populations. So transferability of the findings is very hard, exactly in light of the genetic diversity, which I tried to explain before. Now, the lack of ethnic diversity, the strong European bias, is found in other public genomic databases, not just in databases regarding genome-wide association studies. On the left here, we have the case of GNOMAD, storing a lot of sequencing data from large-scale projects. Again, a very strong European bias. On the right, we have the case of GTEx, the genotype tissue expression database, storing gene expression data from a variety of human tissues. Again, a strong, very strong European bias. Now, the transferability of genetic findings is a real problem. For Mendelian diseases, not much of a problem, because a pathogenic variant usually causes disease regardless of the population in which it occurs. For oligogenic diseases, complex traits, few genes with moderate effect sizes, these are less likely to transfer across populations. And for polygenic diseases, small effect sizes, transferability may be very limited, and linkage disequilibrium will affect transferability. Now, the effect of LD, linkage disequilibrium, on transferability is well explained on part B of the figure on this slide, which has been derived from the paper Sarah Tishkoff, Scott Williams, and myself published in Cell last year. In the case where a single common causative variant is tagged by different SNPs in different populations, well, that's not a problem. Okay, they will all see, these variants here will all see the causative variant. However, if the tagged SNPs derived from a single population, for example, the European population, we go back to the example of the strong bias we have at the moment in databases. We use that tag SNP derived from Europeans to analyze other populations where that tag SNP here only sees the causative variant in Europe, but not the causative variant in Asia or the causative variant in Africa. Then that tag SNP derived from a European population will be totally useless in either an African population or an Asian population. Now, another problematic concept in human genetics is the one of race. Here we have the case of dizygotic twins from the UK in this case, sharing 50% of their genome. Would these girls walk into a healthcare facility? They would be labeled differently. One is a person of European ancestry and the other one is a person of African ancestry. And these categorization may have consequences in terms of diagnosis, in terms of treatment, eventually in the type of medical care given. Now, one way to use ancestry-derived information for medical care is to go through genetic or genomic analysis. And here we have a beautiful example regarding the diagnosis of type 2 diabetes using a standard test, glycated hemoglobin. In African Americans, a gene on the X chromosome, G6PD, may carry a variant at high frequency, which is associated with a decrease in glycated hemoglobin in hemizygous men carrying one copy of the gene on the X chromosome, or homozygous women, 2 copies. And these variant may cause a large number of African American adults, over 600,000 adults with type 2 diabetes, to remain undiagnosed when they are screened with glycated hemoglobin. So the example I showed before was about a variant frequent in a minority group affecting diagnosis of a common disease. It is certainly true that minority groups have been included in large genomic initiatives, large biobanks assembled in Europe and North America, where biological samples are combined with a wealth of health-related data. The problem is that for research projects, when data from minority groups are available, many researchers tend to discard them. And the reason for this is samples from minorities are small, they lack statistical power, they are less likely to allow discovery, and even when a discovery is made, the replication of the finding may be extremely problematic. And then there is a concern of confounding effects. A genetic variant happens to be more common in an ancestry group, which happens to have a higher rate of a given trait, that will cause an artificial correlation between variant and trait. Overall, though, there is a missed opportunity here. Analyzing data from minorities, it's extremely important, both for equity and discovery. At the University of Pennsylvania School of Medicine, there is a major commitment on recruiting minorities in large-scale genetic projects. The Penn Medicine Biobank well represents this commitment. As of 2020, the bank has recruited 65,000 patient participants, all recruited within the Penn Medicine system. Samples from patients are stored in bank. We have a right to recall these patients for further investigations. Samples from these patients are undergo systematic genotyping and sequencing. And the bulk of genetic data is connected with all the health records present in the Pan medicine system. And that provides a huge opportunity to investigate a variety of phenotypes, possibly connected with genetic variation. But most importantly, 20% of participants in the biobank are of African ancestry. Now, the Pan Medicine Biobank is proving to be very helpful in the current COVID-19 crisis, and I will get back to that in a minute. The coronavirus crisis is disproportionately affecting African Americans, with striking disparities in rates and severity of disease. Nationally, 20% of US counties that are disproportionately African American account for over 50% of COVID-19 diagnoses and almost 60% of COVID deaths. There are at play socio-demographic factors, economic factors, racial discrimination, as well as the disproportionate burden of comorbidities, as mentioned before, hypertension, diabetes, and so on. All these contribute to morbidity and mortality. And then West African and African American communities have a high prevalence of mutations like sickle cell disease, like mutations in the APOL1 gene associated with kidney disease. All these contribute to high morbidity and mortality in these communities. Now, it is possible that human genetic variation plays a role in COVID-19 susceptibility, both in terms of morbidity and in terms of mortality. The Pan medicine system is in a position to recruit large numbers of subjects, COVID-19 positives, COVID negatives, broadly across a range of disease severity from asymptomatic, mildly symptomatic, up to patients admitted in intensive care units. We leveraged the Pan Medicine Biobank to rapidly initiate a research program. PMBB is really the backbone infrastructure to recruit positive and negative patients electronically. As I mentioned before, we have currently 60,000 and more participants enrolled in the bank, thousands already with sequencing data and genotyping data available for investigation. And all this is connected to all the electronic health records in the Pan medicine system, from lab data to imaging data. And then, as I said before, we have the ability to recontact. We initiated a study on the host genetics of COVID-19 to identify genetic variants that predispose to infection, identify genetic variants that predict worse outcome, identify genetic variants that are protective from infection. I would like to point out that two very important studies led by Dr. Jean-Laurent Casanova from Rockefeller University, New York, and Necker Hospital in Paris, have just been published in the journal Science. The first study showed that patients with severe COVID-19 may carry rare mutations which affect the production of type 1 interferon, a molecule which is very important to fight viral diseases. The second study found that 1 in 10 of people with severe disease carried antibodies against interferon, blocking the action of the molecule. At the Pan Medicine Biobank, we are taking a broad approach to investigate the genetic basis of susceptibility to infection as well as to disease severity. And in the next slide, I will show what our approach is. So here is how it works. We can investigate sequence variation in genes very relevant to COVID-19. This is the case of ACE2, a main receptor for SARS-CoV-2, the agent of COVID-19, and connect sequence variants with phenotypes, comorbidities that are very important for COVID-19 clinical severity. Here is the case, for example, of viral hepatitis on the left, or it could be the case of respiratory condition, pneumoconiosis on the right. So comorbidities which are considered to be a key factor in determining severe progression of COVID-19. Along the same lines, we can study the possible association between cumulative effect of rare variants, missense or loss of function in key genes, again the case of ACE2, with infectious and respiratory conditions or something else, which are important in terms of progression of COVID-19 disease severity. We also took a population genetics approach by investigating genetic variation in genes that play a role in COVID-19, as that will provide insights into potential differences in susceptibility among human populations. Remembering, however, as a caveat, that non-European populations are unrepresented in the majority of current genetic studies of COVID-19 susceptibility and severity. We have also systematically analyzed ACE2 coding variation in African populations from the large biobank which Sarah Tishkov has put together over the years, and found that there wasn't much variation there other than a few variants, coding variants, present at high frequency in pygmy populations from Central Africa. So most of the coding variants in ACE2 are rare in global populations. Many of them were predicted to be deleterious, indicating that ACE2 is under strong purifying selection. Nevertheless, we identified 4 variants, 3 common missense variants, 1 regulatory variant in pygmy populations from Central Africa. And most interestingly, we detected a signal of positive selection at a region encompassing the 4 variants, indicating that something in that region is there which could provide a selective advantage to those pygmy populations in their environment. Now, could these variants be relevant to COVID-19? We do not know. However, it's certainly worth exploring. And I would like to point out that if we would have focused only on European populations, for example, we would have never seen these interesting signals, which are now opening the door to further investigations. And so the previous example highlights the importance of studying global populations in order to gather as much information as we can and understand how genetic architecture can really underlie susceptibility or resistance to disease. So there are many problems really that result from lack of ethnic diversity in human genetics. It impedes our ability to fully understand the genetic architecture of human disease. It also has an effect in exacerbating health inequalities for populations who are neglected and left behind. Our ability to translate genetic research into clinical practice or public health policy may be incomplete, will be incomplete, and could be mistaken. Attempts to use estimates of genetic risk derived from European-based studies and applied to non-Europeans may result in inaccurate assessment of risk and also inappropriate interventions. And then finally, a point I did not touch on but is important, may result in inaccurate assessment of pathogenic variants in clinical genomic studies. I will finally like to acknowledge and thank my colleagues at the University of Pennsylvania, Chao Zhang, Anurag Verma, Shefali Verma, Yanqing Feng, Marcelo Melo, Marilyn Ritchie, Daniel Rader, Sarah Tishkov, and at Case Western Reserve University, Scott Williams. Thank you very much.